So cohesin plays an incredibly important role in constraining and holding together the two sister chromatids, but we also mentioned a second machine, and that machine is called Condensin. And it's also worth thinking about this a little bit like a rubber band. And so we can imagine, again, I'm going to draw a very similar structure. So these proteins are all interrelated. It's going to be a pair of these SMC subunits, but just different proteins, but same sort of long coil region. There's going to be things that hold this together over here. But instead of the DNA and two different chromosomes going through this, the way that it's going to work is it's going to pull the chromosome through here onto the other side, but come back around with it and essentially bend it over and come back again. So it's entrapping two strands of DNA within the circle, within that chromosome. But instead of one strand of DNA, instead of two different strands of DNA, it's the same strand. So imagine essentially the linear structure. I'm not even sure that this is going to work. But we have something here where that was a fail. Hold on, grab my chromosome back. Don't try this at home. But if we have our linear strand of DNA here, what we're going to do is bring it through that rubber band, but essentially pull it out like this, okay? And so we're holding it together at the base, but we get a loop out of it, okay? And these machines actually are incredibly good. Condensin and cohesin are amazing, not only to entrap, but they can actually move the DNA through this. And so this is going to form a loop. And this loop is actually going to extrude the DNA. And so these machines, actually both cohesin and condensin, they have ATPase activity, and they will actually work a lot like motors to push the DNA through. So if you imagine you have a linear piece of DNA, maybe you start to get to the point where you get a condensin loaded, so that it has a couple of little blips in it where it can again hold down and sort of pinch it at the base. But that ultimately what we're going to do is extrude the loops of DNA through there so that upon a single scaffold, we can make these little different domains. And that's going to allow you really to organize and compact that chromosome in a much more substantial way. So you have these two related but different functioning machines, cohesin and condensin, which are going to help to be able to build up this chromosome, hold on to the pairs of replicated sister chromatids until you're ready to release them, but also compact that chromosome as it goes into mitosis.